Okay, good morning, everyone. I hope everyone's nice and refreshed after the nice meal we had last night. So think I can safe to say it was very enjoyable, and everyone participated and made it very, I suppose, exciting, and a lot of nice conversation was had. So this is, I suppose, our third day here. We've had very exciting 2 days, so let's make today just as energetic, I suppose, as the last 2 days. Again, we'll have a keynote speak, we'll have a tutorial, technical sessions, and then a debate panel at the end of the day. So I'd say it's a very, very nice program that we're looking forward to here. So just one thing as well is I've sent an email to everyone with details on how to give us feedback through a survey. So I'd appreciate if you could have a look at that during the day and get us some feedback, even on the first 2 days, and we could have some metrics and report back to you just to have a discussion on how things are going. And then the overall workshop and feedback on today, we can collect that in the coming days as well. So I'd like to present our third keynote speaker of the event. So we're very happy to have Professor Richard Morris from the John Innes Institute. His speciality is in without any further ado, I'd like to welcome Richard too.
Thank you. Is this working? Yes. So, of all the elements in terms of communication, there's really one that stands out, and that's calcium. Calcium's amazing. Almost every biological process that we can think of is governed by calcium. So, for instance, our vision, seeing, hearing, muscle contraction are all governed by calcium. Also, our heartbeat. Calcium also activates fertilized eggs. So in that sense, we're all the product of calcium waves. Also, when cells die, that's signaled through a calcium signal. So calcium has been termed a life and death signal. And if anyone actually remembers anything at all from this talk, it'll be down to calcium, which is responsible for short and long-term memory. So calcium is really a key messenger in biology. Also in plants, calcium plays a key role in signaling. Many environmental stresses are signaled through calcium. For instance, touch, wounding, cold, heat, drought, pathogen perception are all transmitted through calcium waves. So a key question in both the animal and the plant field is how can such a simple element transfer and control so many different processes? Calcium is a stable ion. It's very stable in the plant. It doesn't change itself chemically. How can that transmit information? So I'm going to focus in on one process today, and that's nodulation. So what you see here are nodules. These are plant-derived structures that form on a group of plants called legumes. These include peas and beans. It's a very expensive process to go through. For the plant is essentially making homes for bacteria. So these nodules here are full of bacteria, and they fix nitrogen for the plant. So we're interested in how that process is controlled. It's an interesting developmental process to think about. But it also, more than that, this process potentially holds the key to sustainable food security, in that plants that form these nodules do not require nitrogen fertilizer. And given that the world's population depends on crops grown with nitrogen fertilizer, understanding this process in such detail that we can transfer it from legumes into crops could potentially be game-changing. So just to talk you through that process. So this is a model legume called Medicago, and if we zoom in onto the root system, what I'm showing here are files of root cells, and plants produce signaling molecules called flavonoids, which they release into the environment. These flavonoids are perceived by bacteria, and that induces the production of another signaling molecule called nodulation factor, or NOD factor for short. And the bacteria release that into the environment, and that's perceived by the plant. And if this chemical communication process is successful, i.e. the right plant finds the right bacteria, the plant then goes through this elaborate developmental process and produces the structures here I talked about before, nodules. And given the title of my talk, it may not come as a surprise that this process depends on calcium. So what you're seeing here is a root hair that's been injected with fluorescent dye that's calcium responsive, and we've exposed the root to nodulation factor. That's the signal that comes from the bacteria that initiates nodulation. And what you're seeing here around the nucleus is this elevation of calcium oscillating. If we block this oscillation either genetically or chemically, we don't get nodules. So somehow this oscillation is transmitting information about the plant, what the plant should do next, and is key for the nodulation process. So the question is, how do these oscillations of calcium concentrations, how do they control transcription, and how do they control development from that point onwards? Now, that's a big question, so I'm going to break that down into 3 short stories that I'd like to tell you today. And they basically map onto the encoding, transmission, decoding structure of information theory. So I'm going to look into how these oscillations are generated, how these calcium signals are transmitted, and then I'll share some ideas with you about how they're decoded. I should say that we don't understand any of these processes, but I think we're making some advances. And what I'd like to do is show you the value of computational biology and modeling in trying to understand these different biological processes. So the first question, how are calcium oscillations generated? So again, big question. Let's break it down into 2 bits. So the first one is called calcium homeostasis. That's basically how does calcium maintain this low level of calcium within the cell? And again, you could come at this from a communications point of view. How do you make sure that you can transmit signals effectively while you reduce the background noise, so you reduce the basal level of calcium? And then we're going to look into how these signals are generated. And it's interesting about how you maintain this basal level, because outside of the cell, the concentrations are around 10 millimolar, and within the cytoplasm, I can see this, they're around 100 nanomolar. So that's a concentration ratio of 10 to the 5. Okay, given that the cell membrane is maybe a couple of microns thick, this is a huge concentration gradient that the cell has to invest lots of effort into maintaining. So this is a very, very low level compared to outside. And the way that this is done is through calcium pumps. We know a little bit about how these work. Just to show you in more detail what this looks like. So this is a membrane, and you have a protein, a very large protein complex, a pump like this. And these are very expensive to run in terms of energy. So 20, 40, 60, 80. Calcium pumps require ATP to function. And what they do is they can pump calcium against this 10 to the 5 concentration gradient. And you can work out how much ATP it takes to do that, and to just take 1 calcium ion against this 10 to the 5 concentration gradient requires exactly 1 molecule of ATP. So it's for the cell, a very expensive process. But again, if you want to build an efficient communication system, it's worth investing in making that noise level low. Furthermore, calcium's toxic to the cell, so the cell does have an advantage by lowering and maintaining this low calcium level. So with that, we have an idea of how the baseline calcium level is maintained. Let's look at the spike generation. So these spikes typically have a very, very fast increase and then a rather slow decline. How are those generated? So because of this high concentration gradient, basically all you have to do is is stop preventing calcium from entering in. Again, the calcium's going to try and cross this membrane just based on the concentration gradient. So calcium channels are basically things that open and close. And if you just open up very, very briefly, this huge concentration gradient will flood the cell with calcium, and then you close it again. So channels, we know calcium channels have to be involved in this process. And then after it comes into the cell, you then just pump it out again. We also know from genetics that there's a potassium channel involved, and we don't know quite what that does. It could, for instance, regulate the voltage across the membrane, or maybe it opens the channel. That will become clear later. But we know that these three components are involved. Unlike the animal field, in these plant components are very ill characterized. We basically know which genes they correspond to. We have no parameters. We know nothing else about them. But what we can do, we can throw these into a simple system of ordinary differential equations, and we can play with the parameters and just see if these components are potentially sufficient to generate oscillations. So I won't go into too much detail. It's a very, very simple model. Essentially, we're just looking at how the membrane voltage varies as a function of the charges that move across, and then we're looking at how much charge goes through these channels. So two-dimensional differential equation. And what we can do is we can then take this equation and we can fit that to the experimental data of the spikes that we've got. So what I'm going to show here is a movie of how that might work. So we've got the three components that I outlined earlier. We have a calcium channel. We don't know how that works. We've hypothesized that it could be voltage gated, it could be ligand gated. We don't know. We've made some assumptions on that. We have the pump that requires ATP and just pumps calcium out of the system. And we have this potassium channel that we don't quite understand what it does. So I'm going to start this movie soon, and I won't go into too much detail, but just what we've done is we've parameterized the positions of these channels based on the result of the ODE. So you'll see these channels opening and closing, and you'll see fluxes through, which are computed from the ODE model. And some bits happen very, very quickly. So in key moments of this simulation, I've slowed down the time step so you can see what's going on. So if I just talk you through it, there's a very quick blip of calcium here. That's sufficient to flip the charge distribution on the membrane. That activates the potassium channel, which keeps the calcium channel open, which means that more calcium can go through, high calcium flux. The more calcium goes through, change of the membrane voltage, potassium channel closes, and the pumps activate and pump calcium out of the cell. So this very simple model is, at least in principle, capable of reproducing these oscillations. And you can see how the components need to interact in order to give these oscillations. And then what we can do is we can take this system and fit it to one individual spike shape. And if we do that, we get a pretty okay fit. So what I'm showing here is the x-axis in time, calcium concentration on the y-axis. This is the experimental calcium trace that you get during the initiation of these nodules. And what we have here is the simulated data. So first observation, the spike shape fits rather nicely. Well, that's hardly a surprise, but that's what we fit to. But if we just fit to the spike shape, we do actually get the right frequency about, right? So that's pretty encouraging. So a very simple model can reproduce these oscillations. But it's a little bit too simple. There's a problem here in that this system is so robust, it will always oscillate with the same frequency. We can't change the spike shape very easily. And there's no way, with the parameters that we have, of stopping or starting the oscillation. But we know that they start upon nod factor perception. They go on for maybe a couple of hours, and then they stop again. So how can we understand initiation and termination of these signals? And you'll remember from Tom's talk on Tuesday, where he showed this picture of all the crowded cell of all different proteins. And many of these proteins can bind calcium. So in Arabidopsis, another model plant, around 500 proteins have been assigned a calcium binding function. So we know that, and also small molecules can bind calcium too. So we know that the cell is full of stuff that can bind calcium. So we've called these calcium buffers. They're just molecules that bind calcium. So if we add that to the equation, so now we have a three-dimensional equation where we just have calcium binding to and from individual buffers. And then we can see how that changes the system. And we see something quite fun there. So if we start with a very low buffer concentration, we have this blue here. That's the stable state. That's just basically a baseline state of calcium. If we increase the buffer concentration, this is all done through a bifurcation analysis, you see that we suddenly have two stable concentrations. So that means it's oscillating. So the system bifurcates at this buffer concentration and goes from a baseline state to an oscillatory state. So that means we have a mechanism for understanding how the oscillations are initiated. You change the buffering capacity of the cell and you can start oscillating. You change it again and you can terminate oscillations. If we look how that looks in phase space, what we can see is that, so this is membrane voltage, buffer concentration, and then the calcium concentration. We see the emergence of these stable limit cycles. So that's a stable oscillation. If we increase the buffer concentration, we see that we transition through phase space and end up with a second stable limit cycle. So we can go from one stable limit cycle to another. And that, of course, if we're going from one to another, that's the change in spike shape that I talked about earlier that we couldn't get with this simple model. If we then have a look at what that looks like in terms of the time evolution of the system, we also see something quite fun. So in the previous color scheme, we had one red limit cycle, and then we were increasing the buffer concentration, now shown here on the x-axis, and then we get this other stable limit cycle here. So these are stable cycles, as in stable amplitude, stable frequency, stable spike shape. But this transition from one to the other, we see this rather strange rapid spiking phenomenon. So if the model's any use to it, maybe this is something that we can see experimentally. If we change the buffer capacity, we should be able to see rapid spiking. And of course, I wouldn't be mentioning that if we didn't have the experimental data to go with it. So if you take a root hair cell of a plant, expose it to nod factor, wait till it oscillates, and then add more nod factor to it. So these are different spiking cells, and the red line here signifies the point where we're adding further nod factor. And what you can see is the system indeed goes through this rapid spiking behavior. So that's indicative of the fact that maybe upon nod factor perception, the cell is releasing further buffer into the system, and that's initiating these oscillations. Furthermore, we can play with the binding constants of calcium to these buffers. And this is taking the binding constants of two different fluorescent dyes that are frequently used in such experiments. And you can see that we can change the spike shape here. So we have a mechanism now of initiating, terminating, and changing the spike shape. And I mean, it's not the best fit in the world, but given that this is a very, very simple model, that's a pretty decent fit to the experimental data in terms of spike shape. Interestingly, until we published this, the idea in the field was that the spike shape is actually characteristic of the stress that the plant's perceiving, and that's part of the encoding of information process. And what we could show quite nicely is after this, that if you cluster all the experimental data just based on their spikes, they fall very nicely into two categories based on the dye that you're using in the experiment and not on the stress. And we can recapitulate very nicely now what the experimental system that you've got. So that was the first story. What have we shown? Well, we've shown that you can understand these oscillations with a very simple model. So we think we know how these oscillations could, in principle, be generated. We have an explanation for the initiation and the termination of oscillations. And we think that we can explain where this spike shape comes from. And coupled with these insights into the system are a number of predictions. So I didn't talk about this in too much detail, but the only way that we could get the model, the oscillatory model working, was to have the potassium pump being activated by calcium. And not just that, we could say how many calcium ions that are needed to activate the potassium channel. And we got the best fit with 2 calcium ions. And this was later confirmed by structural modeling of the potassium channel, which indeed found 2 calcium binding sites. Another prediction is that the potassium channel and the calcium channel, I've written here close together, they have to be coordinated is the main message. So if you introduce a delay between their actions, you don't get these oscillations. So they have to be very tightly coordinated to get these oscillations. But one way of doing that is if they were in close proximity. And that has been shown, and we published that last year in Science. These channels indeed interact, and that's how they function. And we've predicted and verified that you can get this rapid spiking phenomena due to the buffer concentrations changing in the cell. Okay, so we now have some idea, at least for this system, how you might generate oscillations and the nature of those oscillations that you have. And we've also characterized, based on that, a number of the components that we have in the system. So once we have these, how are they transmitted? So this is important because in this image that I showed you, this movie I showed you early on of these oscillations going up and down around the nucleus, all the models that we have are for the cytosol. That's where most mathematical models of calcium oscillations are focused on. But we know that the oscillations are read out in the nucleus because that's where transcription is controlled. And indeed, it's a somewhat controversial area about nuclear calcium signaling, but through recent experimental advances from our collaborators, they have a nuclear localized dye. They can really show that the oscillations are indeed also in the nucleus. So how do you go from oscillations in the cytosol? So there's the cell that will perceive nod factor or whatever will initiate the oscillations. You get calcium oscillations in the cytosol. How do you get those oscillations into the nucleus? So that's the question. And I've kind of drawn them here as 2 different domains. That's not the case. We know that there's something called a nuclear pore complex that sits on the nucleus, and these are huge protein complexes, and this diameter here goes from about 9 nanometers to about 22, 25 nanometers. So small proteins can diffuse freely through these nuclear pores. We think, and following the suggestions from others, that calcium will also simply diffuse freely through. They're huge compared to iron, so we expect calcium to diffuse through. We also know that the nuclear envelope is covered with pores, as shown here. So based on that, ideas that have been published are that, well, actually, you don't need oscillations per se in the nucleus. You generate them in the cytosol. Then the nuclear pore, the nucleus covered with nuclear pores, lots and lots of holes, calcium will diffuse through, and what you're seeing on the inside is simply diffusion of those spikes on the inside. Makes total sense. So that's kind of the idea that's out there. So then we thought we'd have a look at that. So this is a very simple model. There are 2 spheres. This is now a 3D simulation of calcium. So we've got the components on the outside of the nuclear envelope, not on the inside, but we have several thousand of these nuclear pores that sit all around the nucleus, and we're going to look to see if we can recapitulate these oscillations on the inside. So the framework that we're using for this is called the fire-diffuse-fire model. The concepts are quite simple. You have calcium in the cell, you have channels. If the calcium reaches a certain threshold, that channel will fire. It will then diffuse, and once calcium reaches the threshold at another channel, it will fire as well. And you can write down the PDE for that, and if you choose your geometry carefully, that's why we've got spheres, you can actually solve this through Green's functions. So there's an analytical solution to this. So it's still very, very compute intense to to solve, but basically it boils down to tracking which channel failed at which point in time, and then from that point in time, summing up the analytical solution that's shown here, and that gives you the overall calcium concentration. And that allows us to do some very interesting studies of how these might be transmitted. So throwing all of that together with the channels just on one side, I'll show you what this looks like. So what you'll see here is a simulation of this fire diffuse fire model on the outside, and then you'll see what happens on the inside. And these are atlas views of the surface. So we're seeing the whole surface at once. Okay, so this, these files are absolutely huge. So I've sort of only showing a snapshot here. But what you see here, or what you saw here, was the initiation of a calcium wave. All the channels are firing, they're coordinated, and you'll get oscillations. What didn't happen here, there are individual sparks. We didn't get the oscillations on the inside. And that's puzzling, right? Given how big these channels are and the diffusion, you'd expect to see oscillations here. So we pushed that harder. So there are about maybe 1,000 to 2,000 pores on the nuclear envelope. We increased that tenfold, and we increased the diameter. We still couldn't get oscillations on the inside. So you have to be somewhat careful interpreting negative results like that, but we did try very hard to make that work, and we didn't, and we couldn't. So what that suggests is that the machinery isn't just on the outside, it's also on the inside. The nucleus has its own set of calcium machinery. And indeed, we could show that through antibody and immunogold labeling. We could show that the components are roughly 50-50 distributed between inside and outside, and we published this a couple of years back in PNAS. It's the first demonstration that the nucleus has its own calcium signaling machinery and can do both. It can get both synchronous and asynchronous oscillations through having these different machineries on both sides. So if you have them on both sides, why do you have them on both sides, right? You only actually need them in the nucleus where you're decoding calcium oscillations. So this is summarizing a huge amount of work in just one slide, but basically what you've got is you've got two coupled oscillatory systems. But more than that, so this is showing two channels, A, D on the inside, for instance. Now, it might be that these channels are so far apart that diffusion is unlikely to go from A to activate D. So that means that you get one spike at A, and then the oscillation would die out. But if you have nuclear pores and channels on the other side, what you can get is a situation where you release calcium here, it diffuses to the other side of the nuclear envelope, and then it makes its way through the channels on the other side, and then gets, so via B and C, and then gets to D and initiates it. So you're opening up a whole new set of routes from one channel to the other. So what this means is that the oscillations become a lot more robust to fluctuating environments. So that means that you can sustain the oscillations for much longer. And that, I'll get onto, that will tell us something about what the decoding mechanism might be if you need a lot of oscillations. So that's interesting. So, and I should say as well, they're coupled oscillatory systems, but they don't have to have the same frequency. So again, this is quite a controversial area. Some, with the main idea being that they're just diffusing through the nuclear envelope. We've shown that isn't the case. You can get oscillations that have the same frequency and with a minor time delay, and you can also get different oscillations. And that fits with this model of having the machinery on both sides, and depending on the strength of the coupling, i.e. how much you open or close the nuclear pores, you can get different or similar, or sorry, different or same frequencies. Okay, and it provides a robust way of maintaining the oscillations. So what do we learn from that? We've learned that diffusion through the nuclear pores is unlikely to be sufficient for the transmission of these oscillations. And we think we have an explanation for why these spikes can go on for several hours due to this coupled system that we have. And predictions from that. So we think that we predicted the calcium machinery should be on both sides of the nuclear envelope, and indeed that's been shown in the meantime. And if the cell's going to the effort of making sure that these oscillations are robust to sustain them over a long period, well, maybe the number of oscillations or the sustained length of oscillations is important for signaling and decoding. And that brings me on to the last story I was going to tell you about, which is how the oscillations are decoded. So we'll break this down again, looking at this these typical spikes, so baseline and this spiking regime. And one simple way of looking at this is there are simply two states. There's an off state, so that's the protein sitting in the cell, baseline calcium, doing nothing. That's off. That's easy to understand. On. So then protein sits in the cell and you get this wave of calcium going up and down. How does that activate the protein? So that's the question. So off, I think, is clear. How do you activate protein through these spikes? How do you decode that information? Well, so we can explore a few ideas how that might happen. So one is conformational change. So we know that calcium binds to proteins, and we heard from Tom's talk as well that binding of things to proteins can induce conformational changes. We know this is going to happen. Here's a canonical example it's called camodulin. So you have, on the left, you have the sort of folded up structure of camodulin without calcium, then you bind calcium and you open up like that. And in that open form, camodulin can bind to another protein. So it's kind of like an amplifier, right? It's taking a small change of something to something, a next step up, where it can bind or activate a protein. So that's certainly a way that you could activate a protein with calcium. Is it the only one? Well, we also know that proteins have internal modes of vibration and can go through internal conformational changes. So maybe, apart from the binding, maybe could be some kind of resonance effect of these oscillations driving these internal vibration modes of the protein. It's an idea anyway. What else could it be? So maybe protein, well, I've got no idea how, but maybe the protein is actually counting oscillations. 1, 2, 3, and then it needs a certain amount before it's activated. So those are at least a number of somewhat plausible hypotheses, at least for how the decoding might happen. So before I move on, let me tell you something about the oscillations. So we know that they base level, where we said before, around 100, 150 nanomolar, and they go to just under 1 micromolar in spikes that I've shown you quite a few times already. The period is around 100 seconds. So internal modes of proteins are often the sort of pico, depending on the size of the domain that's been moving, to microsecond to, in extreme cases, maybe micro to milliseconds. So it's several orders of magnitude of the frequencies we've got here. So it makes this resonance idea, I think, rather unlikely, right? We're several orders of magnitude off. So let's jump from that into what we think the decoder is, because we know oops, that's come out well. So we know genetically where these oscillations are decoded. So there's a protein called CCAM-K, and that sits downstream of the oscillations, and we know genetically that that's likely to be the decoder. And if we look at this protein, it has 3 domains. It has a kinase domain, so something that will phosphorylate another protein. It has a camodulin binding domain. So camodulin is this protein I showed a couple of slides back that binds calcium as well. So here's a image of camodulin. These yellow bars here are called EF hands. These are protein motifs that bind calcium. So essentially, the calcium typically binds to oxygen. It can be coordinated 4 to 6. So they're basically hands that grab calcium like this, EF hands. Those will bind calcium. There's that. That's mine, yeah. So this protein has 3 EF hands, so it can bind calcium. That's good. Maybe it is the decoder. Furthermore, it can bind calcium indirectly through chemodulin. So lots and lots of potential for calcium affecting the structure. We don't have a structure, but we built a homology model of what we think it might look like. So the kinase domain is pretty well defined, and the rest is lousy, basically, because of the lack of homology with anything else that we know. Kinase domain, chemodulin binding domain, and this domain is called the vicinin-like domain with 3 calcium hands. And this protein is unique to plants, and it's unique to a certain type of plants, namely these legumes that go through this symbiosis. Okay, so we also know that phosphorylation that the protein can be phosphorylated, and this phosphorylation event is key for the activation of nodules. So how might that work? So I'm not going to go into this in a huge amount of detail, but just say that our collaborators went through a huge amount of scanning through this protein, looking at various mutants, various deletions, and seeing what the effect was. So there are deletions and mutants all over the place that, at this point in time, make no sense at all. But what's interesting is that you can turn the protein on by some mutations and some deletions. So that indeed suggests it might be the decoder. You can actually turn this on. And what you're seeing here is the experimental readout. These are spontaneous nodules. So these happen without the bacteria, without calcium. Something that's been done to the protein is switching it from this off state to the on state. And without anything else there, you're getting these nodules. So that's just supportive that this protein might indeed be the decoder. And we have a number of different data that we'll need to reconcile with whichever model that we come up with of how this might work. So we know from the work of others that in order to activate the protein, you need to break a hydrogen bond. So that's shown here. So kinase domain, chemodulin domain, and these domains are stuck together with a hydrogen bond that you need to break to activate the protein. So we basically have no clue what that does, but a plausible hypothesis could be that if this is the active site of the kinase, that has to be exposed for the kinase to be active. Maybe this huge other bit of the protein is sort of bent round, covers the active site, therefore it's inactive. You break the hydrogen bond and it swings round. You expose the active site, it becomes active. So that's kind of the idea of how it might work. And if we zoom into this homology model that I built, you can see nicely this hydrogen bond between 2 residues, one on this sort of linker domain here and one in the kinase domain, and there's this hydrogen bond here, and that's what we have to break to activate the protein. And we'll hear a bit more about that soon. So we know the protein, we know that phosphorylation is important, we know that we have to break a hydrogen bond, and we know that it's calcium activated. So again, we don't know quite how that works, but one hypothesis is here are 3 calcium binding sites. Maybe calcium binds to these EF hands. The binding energy released from that will break the hydrogen bond, and so calcium binding to this, conformational change, and swings round, protein's active. So that's a very attractive model, and would make sense, fits with similar-ish models that you find in the animal area. So with a mechanistic model at our hands, we've determined what the binding constants of these EF hands are, and got a surprise. So again, calcium basal levels are around 100, 150 nanomolar. One of these binding constants is 20 nanomolar. So that basically always has calcium bound. It's really, really tight binding. And the other 2 are in the range of 200 nanomolar. So that's about basal level calcium. So what that's suggesting is that actually, even without calcium spikes, this thing is binding calcium. So now we have binding constants, we can throw that into a very simple steady state model of what the different species are. We can take phosphorylation rates from animal systems, and we know the concentration of camodulin, and we can guess the binding constant. We can look at these different concentrations. So camodulin just by itself with nothing bound is what you probably just can't see here. So unless you really have no calcium in the system, that will not be a dominant state. Once you have calcium in there, the tight binding site will always have calcium, that will always bind, and then even at low calcium levels, the state without calcium bound basically won't exist. What turns out to be the dominant form at basal level is actually the phosphorylated form. So this goes totally against how we think other systems work, where phosphorylation is the activated mode, and against previous hypotheses about how this decoding might work, where I told you about calcium binding, conformational change, and then autophosphorylation sticks it in the on state. And we heard yesterday in the plenary talk about how phosphorylation can go through this bistable state. That's the model that we had in mind. Maybe we can do that. That's exactly not what's happening here. Phosphorylation seems to be happening already at basal levels. But the higher you go with calcium, the more you get of this camodulin binding to the phosphorylated state. And interestingly, and this isn't fit, this is just based on experimental data that we have, this switch over here of which state becomes more dominant is at the amplitude of the calcium oscillations that we've got. So it looks like we have an off state which is phosphorylated CCAM-K. You increase calcium, and then you get this state becoming more dominant. That would suggest that's the off state, that's the on state. Okay, so could phosphorylation be the off state? Well, let's have a look at this hydrogen bond again. So if this is phosphorylated, what happens? So we know we need to break this to activate the protein. So other groups have looked into this, and they phosphorylated this protein in silico. And here are two domains that are close together, hydrogen bond. You bang in a big phosphate, that's a big bulky thing, that pushes the domains apart, breaks the hydrogen bond, activates the proteins. So many, many moons ago, I worked on and solved protein structures, and that to me didn't make any sense. So phosphate is covered with oxygen atoms that will very, very readily form hydrogen bonds to anything that it can see. So although it's an attractive model in terms of bulkiness and pushing apart, didn't fit with what I thought should happen. And it turns out that if you do this modeling, all the default parameters for force fields in molecular dynamics simulations do not have phospho residues included. They only have standard residues included. So a little bit of effort, but you can include phospho residues and include the parameters in the force field. And if you do that, suddenly the domains aren't pushed apart, as had been published, they're pulled together. So the phosphate's holding onto the domain, and you get these additional hydrogen bonds. It's very stabilizing this off state. So the phosphate doesn't break, it reinforces this hydrogen bond and really locks it into this off state. So it's a very stable off state, and that makes sense. The cell doesn't want to make nodules if there's no bacteria there, and you would want to lock it in and then override it. So the hypothesis there is that you need to phosphorylate to power off and turn off this protein. And again, that fits. So we now have this plot I showed earlier, which was the first indication that the phosphorylation state might be the off state. We have the homology model where the hydrogen bonds fit as well. And interestingly, if we go back with this through all those different mutants that I showed, everything fits. If you can't phosphorylate that residue, you can't stick the bonds together. That's when you get spontaneous nodules. If you change that threonine for an alanine or something that you can't phosphorylate, you get spontaneous nodules. If you break the EF hands, that also break these bonds, you get spontaneous nodules. So all these different mutants that looked like they made no sense fit with this model. Okay, so how might the decoding work? So we can throw everything that I've told you about into this time a rather large set of ordinary differential equations. Essentially, we're just modeling all the different species and calcium coming on, off, and camodulin binding and phosphorylation. And we know some of the parameters. We know the KDs. We don't know any of the on and the off rates. We don't know any of the phosphorylation rates, but we can make guesses as to what they might be. And then we can simulate the system and see what happens. So this is a bit complicated, but I'll talk you through it. So in black are simulated calcium oscillations, and in blue is the phosphorylated form. That's the dominant form when we have no oscillations. And what the oscillations do is they're taking away from this off state by binding to camodulin. So what you're seeing here is that at high calcium concentrations, camodulin is being activated through calcium. That's binding to CCAM-K, and that's then the active form. And that's what you see here. But interestingly, it's not this on or off state that we and others thought was going to happen. The activated state basically just follows the oscillations, right? It's like constantly a buzzer or something, constantly pushing. So there's no on or off state as such. There's an active form that is conditional on the calcium being up, and then it goes down again. So in that sense, there's no memory in the system. But if we look at what CCAM-K does, so that has a kinase domain that will phosphorylate another protein, a target protein. So if we monitor what happens to that protein, that's shown here in magenta. And that so every time it gets active, it will phosphorylate. So the population of phosphorylated proteins will increase over time. So actually, that's where the memory resides in the target phosphorylation, and that goes up over time. And so that's where the on switch is. Okay, I've talked a bit longer than I thought. I will summarize that very, very quickly. We think that calcium at basal levels turns the protein off, not on. We think that at high calcium levels, this is overridden by camodulin binding to the system, and that then phosphorylates its target. So getting back to the three hypotheses, is it conformational change or resonance or counting? We have this strange result. Well, actually, it's all of them, because calcium is binding to CCAM-K going through a conformational change, binding to camodulin going through a conformational change. Resonance, I almost excluded a priori because of the timescales. But if you look at the whole system as a system, there is a resonance there in that if you have the dephosphorylation rate, as long as that's about the same order of magnitude as the driving oscillation, that's when you can get this saturation and sort of ratchet effect of increase in target phosphorylation. So you could argue there is some kind of resonance in the system. Counting, well, how on earth could that work? Well, actually it does, because every time you give it a calcium spike, you increase the phosphorylation of the target. So actually, you could argue it's counting spikes until you reach a maximum phosphorylation level. So actually, we think it's all of those things together. Okay, so what is the modeling for the decoding shown? So we think that we have a model that shows us how oscillations might be decoded, and it explains all the mutant data that we have to date, which I think is encouraging, at least. It makes a number of predictions. So it says that if we opened up the cell and looked at CCAM-K, this will predict that the sort of natural default form of CCAM-K is the phosphorylated form. And it would predict that the phosphatase activity needs to be slower than the oscillations. That's this resonance effect that I told you about. And it would indicate that the number of spikes that you get would lead to different phosphorylation levels. So unlike the other predictions that I talked about earlier, where they were all validated, none of this has been shown yet. So the jury is really still out on this, and probably it's all wrong, and that's fine. Every model is wrong, but at least it gives us a framework for thinking how this might work. Okay, with that, I'll draw to an end. I hope I've tried to convince you that calcium's a fun thing to work on. I've hoped I've shared some of my excitement for calcium, and also conveyed that computational methods have a key role here to play in trying to elucidate the process and understand the mechanism of what's really going on in this system. Okay, and so just a final word. So everything I've talked about today was intracellular calcium. I'm just going to end by showing you a movie. This is something that we worked on recently. So this is a root of an Arabidopsis plant. What we're going to do is going to give it salt stress about here. And then what you'll see is that this salt stress initiates a calcium wave traveling at around 500 micrometers per second through the whole plant. You're seeing this calcium increase in red, goes in all directions, and this calcium wave changes transcription in the leaves. So it's a systemic response. So how does that work? That's the question I'll leave you with. And with that, I will thank Alan for inviting me to what I think has been a very stimulating and fun conference. There's been some great talks, great discussions, and I wish you all the best for the field for taking this forward. And I'll thank my collaborators, and I will try to answer any questions that you might have. Thank you.
So we'll take any questions if there's...
So in this protein structure you showed, why do you still need to bind them to this other protein? So you have the CAM-K is the big protein, and you have to bind to the
Camodulin.
So why do you still have to bind to the camodulin in the end? Why is it there? Why do you need it?
So we think that's the activation mode. We think that it's the off state is when it has calcium bound unphosphorylated. And to override that, to switch it on, camodulin binds, and we think that camodulin then actually breaks these hydrogen bonds of the...
So there's no dephosphorylation going on then? So it's not that there's another protein that dephosphorylates that area to actually open up, because it's removing a very strong binding inside the protein structure.
Yeah, so it could be that you have a phosphatase that takes it off. And in the simulations, that's difficult to reconcile, because then you'd have this constant balance of phosphorylation and dephosphorylation, and you couldn't keep the protein stably in the off state, which is the phosphorylated state. And experiments show that if you then add camodulin to the phosphorylated state, it doesn't dephosphorylate, the phosphate stays on there. So the simplest model for that is that camodulin simply binds the phosphorylated state, but essentially rips these domains apart and activates the protein. So you don't need to phosphorylate first. But the fact that it is phosphorylated enhances the binding constant of camodulin to the protein. So what it's actually doing is it's sort of preparing the protein for an incoming calcium wave, and then it will bind much, much tightlier than without the phosphate.
And then the second question I had was, okay, the protein itself can also bind the calcium. So you have the camodulin has to bind, which contains calcium, and the protein can bind calcium. Why do you need both? I mean, is there a transfer of calcium going from one to the other? Or is it simply there? But I don't see why it would be there for both cases.
I don't have a good answer to that. So the calcium binding to the CCAM-K, it promotes phosphorylation, but it's blocked in the off state, right? So you could put it in an off state without calcium bound, and it should work equally well. So there's clearly something we don't quite understand yet.
But it could be that in part of the decoding that you would have a transfer from the small molecule binding the calcium to the bigger molecule, and actually helps in breaking the hydrogen bonds and actually...
Yes, so that's a good idea. So it might be that it would keep calcium trapped in the vicinity to sort of prolong the active state. That's a possibility. We haven't included that in the modeling.
Thank you. Thanks a lot, Richard. Very interesting. So a possibly naive question, but coming from the communication engineering standpoint. So in the first part, when you were talking about generation of the oscillations, you were talking about a local effect.
Yeah.
And you described that with lumped equations, a local effect, this channel pump combination. Then, when talking about transmission, it was more of a global distribution effect, the firing diffusive fire effect. in either case, a fundamental question from communication engineering would be, are they self-sustained, distributed passive effects, or is there any global orchestrator actively orchestrating these processes?
I very much doubt there's a global orchestration going on. I think this is all based on channels and pumps acting together in concert and giving rise to this. But it would be fun to, I mean, what we've talked about for a while and haven't done, it would be to sort of take that idea and say, and it links somewhat to Tom's question, how would you design this a priori without without having to take into account the evolutionary history of how it's there, so to speak? Could you think of something along those lines of how would you design it globally to work, and what would the consequences be for the local components? And you probably indeed lose the other EF hands, and you'd probably see all kind of other fun things. I don't know. Good question.
Thanks a lot.
So you discussed calcium oscillations in the cytosol and in the nucleus. So based on what you've studied, are there differences in how it's decoded in both of them? Because in cytosol you have signaling, in nucleus it's more genetic based or epigenetic based. So is there any insight on that?
So we haven't looked at decoding these oscillations in the cytosol because we know that the protein sits in the nucleus. So I don't know. So it links somewhat to this specificity question, which I didn't allude to too much, but that's a key question in the field. If you have calcium changes, how does that signal select just one protein to activate? And it's not known how that works. So we've just looked at one protein, so we've kind of dodged the question completely. But you're right, in the cytosol, these oscillations will also be doing things. There are tons of calcium decoding proteins in the cytosol. One question is, why are they not activated if they're not activated? And if they are activated, what are they doing? And we don't know, basically. It's a good question.
Because it's like when you have the oscillation of cytosol, is that somehow, it's one thing that it just diffuses or there's some method, it's a nucleus, but if there's some protein that's activated which now can go through the nuclear pore complex because of the calcium oscillation. And the other is, you mentioned earlier something about cell death.
Yeah.
With too much calcium or some offset. So how does that factor into all this? Like, where do you get that upregulation, I guess, of calcium, which leads to that?
So calcium is a very active ion. It will bind to phosphate groups like ATP, and so that's the reason why it's toxic to the cell. And that's, I mean, evolutionarily speaking, that's probably how this signaling system evolved. Calcium is toxic to the cell, therefore the cell is putting a huge amount of effort into getting all the calcium out. By doing so, it has a system that has a very, very low level, low noise level, but it also has efficient systems of quickly letting in and getting calcium out again. So the signaling is probably a consequence of these toxic effects. So, and if you don't control that, calcium will go up, and then basically you're swamping or killing all the ATP in the system, and the cell dies.
So then a question comes to how long, if this, let's say the oscillation you prolong over time, does that have the same effect as, getting cell death?
So these oscillations can last for several hours. So I think these small blips that can be tolerated. I think if you had a higher sustained level, that would very swiftly lead to cell death. But I don't know how long, to answer the question.
You showed the mechanism on the membrane of the nucleus, where you had these two types of receptors, one tunnels, one outside and one inside, and then you had these spikes going from A to B.
Yes.
And the spikes from B and C were much smaller in amplitude. Why is that?
So that was just one of the simulations that we had. The reason, so that came about, so we looked into different diffusion constants on either side of the membranes. What we were looking at is how far can you push the environmental parameters of either side and still get oscillations or not. So this was the case where you have I'll get it the right way around, very, very low buffering on the outside and a larger diffusion constant, and a very high buffering on the inside with a very low diffusion constant. And a very low diffusion constant would mean that you can't get from A to B because it'll be swamped up by buffers. So this was just a scenario where we were looking into how do you kill this oscillation? We increase the buffer capacity, but by doing so, you also decrease the amplitude that you're reading out. So it was just one of those things we were playing with. It's a reflection of the diffusion constants and the buffering capacity of the system.
Okay, if there's no further questions, I'd like to thank Richard again for a very inspiring talk. So we'll break for a coffee break now, and we have tutorial back here at 11 o'clock. Before then, what I'd like to do is a group photo. I think maybe we could have out on the stairs, if everyone kind of stands on the stairs, and we'll take the opportunity now while everyone is here before anyone else leaves. So maybe we can just go out and try to get that done. Yeah. Thank you.